everyone. Good afternoon. Welcome to Osteo Bites. My name is Christina Iptoma, and I am mom to Osteo Angel Dillon and Director of Scientific Programs at MIB Agents. And today on Osteo Bites, we are talking with Dr. Claudia Benavente about UHRF1 as a therapeutic vulnerability in osteosarcoma. Thanks so much, Dr. Benavente, for joining us on Osteo Bites today. We are thrilled to have you, and thank you to our panelists, Vicky and Walker, for joining us today. Both Vicky and Walker are osteo warriors, and they are also respectively the president and VP of our MIB Agents Junior Advisory Board this year. A little bit more about our guest today. Dr. Claudia Benavente studied Molecular Biotechnology Engineering at Universidad de Chile, where her interest in pursuing cancer research first started. And to further her studies, she came to the U.S. to pursue a doctoral degree in cancer biology at the University of Arizona as a Fulbright scholar. She then moved on as a postdoctoral fellow to St. Jude Children's Research Hospital in Memphis, Tennessee, where she became familiar with childhood solid tumors. Dr. Benavente is currently an associate professor at the departments of Pharmaceutical Sciences and Developmental and Cell Biology, and she's a member of the Chao Family Comprehensive Cancer Center at the University of California, Irvine, which, by the way, I just recently learned their mascot is an anteater. Dr. Benavente's research focuses on understanding how pediatric tumors form to design new ways to treat them. Her research aims to understand how genes are normally controlled in developing tissues and how epigenetic processes are perturbed to facilitate cancers to arise. This information guides her in the development of new therapies. Welcome, Dr. Benavente, and welcome everyone to joining us today. And everyone, please feel free to add any questions you have for Dr. Benavente throughout the talk to the Q&A, and we'll make sure we ask those. And before we get started, we have a couple reminders about Factor. Our Factor conference registration is now open. It's June 22nd to 24th in Atlanta. And there's really something for everyone. We'll have scientific sessions. We have a wellness track for our families. We also have a Warrior HQ that is free to warriors and siblings 18 and under. Some of the panels that we have this year are biomarkers for risk stratification, preclinical models in osteosarcoma, comparative oncology, local control, immunotherapies, and molecularly targeted therapies. And this year, something new, we have a few smaller breakout discussion groups. We have 3, and the topics are researcher's best friend, can dogs expedite bench to bedside for pediatric patients? Also a discussion group on leveraging computational biology to accelerate discovery in osteosarcoma. And then our third group, Unleashing the Power, Patients with Osteosarcoma Who Enable Research. So some really great opportunities to have kind of a smaller group discussion. We also have a limited number of Factor Travel Awards that are sponsored by our MIB Agents Family Funds. And so these are awards up to $2,000 to cover registration and travel expenses to Factor. Special consideration is given to young investigators and attendees from outside the U.S., but any research scientist or clinician is eligible. So please do consider applying. The deadline to apply is next Friday, April 7th. And I'd like to thank the sponsor of this episode, the Osteosarcoma Institute, OSI. They're a nonprofit organization led by osteosarcoma experts from top U.S. cancer centers who together are concentrating on the cure for osteosarcoma. The mission of the OSI is to dramatically increase treatment options and survival rates in osteosarcoma patients through identifying and funding the most promising and breakthrough osteosarcoma clinical trials and science. And in addition to advancing research, OSI also provides a free resource called OSI Connect for osteosarcoma patients. Their osteosarcoma experts can discuss available treatments, possible side effects, and provide helpful advice for getting the most out of your visits with your treating physician. This resource is available in English and Spanish and aims to help patients and families find answers to their questions. So thank you so much, OSI, for sponsoring this episode. Vicky, could you go ahead and introduce yourself?
Yep. Hi, everyone. My name is Vicky Foy, and I'm the president of the Junior Advisory Board this year. I'm an osteo warrior who finished treatment in May of last year. So it's almost coming up to the 1-year mark, which is kind of exciting. But on to you, Walker.
Hi, my name is Walker. I'm the vice president of the Junior Advisory Board, and I finished treatment, I guess it was in spring of 2019, and haven't had anything since then. And then Dr. Benavente, you can take it from there.
Thank you. Thank you for the invitation to participate again. I have participated in the factor meeting a few years back, and that was fantastic. I think it's a great opportunity for families to participate there. Today, I'm going to share what we've been up to in terms of our work. And so, I'll be talking about particular our findings on UHRF1 and its potential as a therapeutic target in the treatment of osteosarcoma. And this audience is no stranger to osteosarcoma, but I do want to emphasize, basically, if you look at the survival rates of osteosarcoma, it really explicitly shows that we have this pressing clinical need to understand the factors that are responsible for metastasis in osteosarcoma in order to facilitate the development of new therapeutics, as metastasis is the main driver of the poor rates of survival in patients afflicted with this disease. And osteosarcoma tumors are highly heterogeneous and present what we typically refer as genomic instability, so a lot of mutations and a lot of chromosomal rearrangements. However, several studies that have done whole genome sequencing have identified recurrent mutations present in a lot of the osteosarcoma tumors. Nearly all osteosarcomas present mutations in the p53 tumor suppressor gene. And there are several other mutations that occur at lesser frequencies, but quite frequent. And I just want to bring your attention to this another tumor suppressor gene called RB1, or retinal blastoma susceptibility gene, that's frequently associated with increased metastatic presence in poor clinical outcomes in patients with osteosarcoma. And I have been studying the RB1 gene for the past 15 years in the context of a completely different childhood cancer called retinoblastoma, in which the vast majority of them present with mutations in RB1. And through the study of RB1 and how in the pathways that RB1 controls to aid in the retinoblastoma tumor formation, is that we identified the target that we were going to be talking about today, UHRF1. And we have evidence pointing out that UHRF1, it's an interesting druggable driver downstream of RB1. And this includes osteosarcoma. So why does this UHRF1?
Sorry, can I ask you a question back on that last slide? So
Sure.
Of those mutations, do you find that they tend to occur in groupings, kind of? Like if you have a TP53 mutation, are you likely to have an RB1? Like, do some of them partner or go together? I guess is one question.
Yeah, so they're not mutually exclusive. P53, I mean, for sure, the example, because there are 95% of patients present with P53 mutations, quite often you would get others. So this is sort of like the requirement, absolute requirement for osteosarcoma to form. But you will get P53 and RB1 mutations. MDM2 tends not to co-present with P53 because they're sort of like targeting the same pathway. But yeah, you can have several of these mutations happening at the same time. And so you can start on appreciating the complexity that then it comes to treating osteosarcomas, because it's not the same to have this mutation, that mutation, and having all mutations. And so the therapeutic opportunities really depend on which gene are driving, what gene mutations are driving tumorigenesis. So I hope that addresses your question.
And just a basic question about RB1, because it stands for retinoblastoma, which I think is so interesting because I haven't really met another gene that's named after
the disease that it was identified.
Yeah, but it's present in lots of cancers, right? I mean, it can be a driver in other cancers. I was just curious, why is it called the retinol?
So this is the very first tumor suppressor gene that was identified. And it's, I think that these also emphasizes the importance of understanding, one of the problems that we have with osteosarcoma is that fortunately, very few patients are afflicted with the disease. They're rare diseases. But not because they're infrequent means that they're less important to be studied. Retinoblastoma is even less frequent than osteosarcoma. But through our study of retinoblastoma, we have identified and learned a lot about retinoblastoma susceptibility gene RB1 as a tumor suppressor. And so the same way, there are many things that we have learned from the study of osteosarcoma that are can be extrapolated into other cancers. So this tumor suppressor, the reason why it's frequently mutated in a lot of different cancers, RB1, and sort of what led me to start studying osteosarcoma is patients with hereditary mutations in the RB1 that survive retinoblastoma quite frequently go on and develop osteosarcoma in their teenage years. And then they're high at risk of developing other cancers through the rest of their lives. And that is because the RB1 gene is a critical regulator of cell cycle. And one of the main characteristics of cancer is that they have lost their ability to control cell division. So every single cancer, in one way or another, either through the direct mutation of RB1 or through other mechanisms, it needs to lose control of the RB pathway in order to those cells that now to be continually proliferating. So that's also true for things like p53. P53 as a guardian of the genome. You've probably heard about this a lot of times because it's frequently mutated in osteosarcoma as a tumor suppressor. It's critical to control cell death. And so you have to, P53 quite often is mutated in order to allow cancer cells to survive. And if you don't see mutations in P53, quite often what you see is amplification of things that regulate the P53 pathway, like MDM2 or MDMX. So they're common denominators across all cancers. And this is why you see frequent recurrency of particular tumor suppressor genes present in several different types of cancer.
Thank you.
Is that good? Okay. So what is this UHRF1 protein? I'm not going to touch too much on the scientific side of things, but in normal cell, so UHRF1 is a protein classified as a ring finger E3 ubiquitin ligase. And what its main role in normal cells is to maintain DNA methylation and the formation of heterochromatin while the cell is undergoing DNA replication. Basically, the cells have a memory to keep and remember what their identity was. In UHRF1, any genes that are permanently shut off, UHRF1 as the cell is dividing, passes on that memory of the genes that need to maintain to be in an off position. So it has been speculated in cancer as to why would high levels of UHRF1 be present. And we can observe several different things, and we can speculate, and you have high levels of a protein that is normal regulating turning off of genes, that maybe if you have a very high levels, now it would go beyond its normal duty of maintaining the off genes into now starting to turn off things that shouldn't be off. And if it's turning off tumor suppressors, the genes that their normal function is to prevent tumors from forming, but that may lead to a disease state. And so there's a lot of roles of this protein, including functions in DNA damage repair in the P53 dependent pathway, and as well as regulating cell division through binding to RB1. So now you start seeing some common denominators with RB1 and UHRF1, but in particular, what I want to emphasize is link of RB and UHRF1 being twofold. One is that we identified UHRF1 as a protein that is directly regulated by the RB pathway. And so you could see, so if RB will normally be working to repress the expression of UHRF1, and you would only want UHRF1 to be expressed while the cell is dividing, so that chromatin can get inherited. So the epigenetic aspect of the cell can be also passed down to the daughter cell. If you lose RB or you inactivate the RB pathway through mechanisms that hyperphosphorylate RB, then you'll see an increased level of UHRF1. Now, what is also being described is that UHRF1 itself can also directly interact with RB and participate in some of the regulatory functions that RB has in the cell in terms of silencing repeats and committing to sulfate lineages, setting up the chromatin structure and DNA repair. So the RB pathway is both directly regulating the levels of UHRF1, and then UHRF1 is interacting with RB to preserve cellular functions. And so what we observe in terms of osteosarcoma in the clinical presentation of UHRF1 in tumors is that when you see high levels of UHRF1 here in the line in blue, compared to patients that have low levels of UHRF1 at the time of diagnosis, then there's a direct negative correlation with patient survival. Basically, having high levels of UHRF1 is predictive of poor outcome in the patients at the time of diagnosis. So we seek out to see exactly what was UHRF1 doing. And one of the very first things that surprised us was that while we had been studying UHRF1 and we were curious about UHRF1 in respect with cancers that mutate RB1, well, we identified as, this 143B, SJS1, SALS2, U2OS, these are all osteosarcoma cell lines that we commonly used in a laboratory setting, and mesenchymal stem cells, like normal cells that give rise to bone tissue. And you can see that regardless, all of this have very, this four cell lines really depict the genetic heterogeneity that we see in osteosarcoma patients. The only one that has a mutation in RB is SALS2. And so regardless of the background, all of these osteosarcoma lines have high levels of UHRF1 and much higher than the normal highly dividing cells that form the tumor. And even when we look at patient-derived xenografts, so these are basically patient tumors that now have been grown in mice, you can see that there is high level of expression of UHRF1 protein in all samples, pretty much. And so to really understand what is UHRF1 doing in the context of osteosarcoma, we quite often make use of genetic engineered mouse models. This is particularly important and useful in developing and studying disease, particularly when we're talking about rare diseases, right? We have very limited resources in terms of patient samples because fortunately, there's not a lot of patients with osteosarcoma. So being able to model what happens in human using mouse model really allow us to understand the biology. And so here you can see what I told you briefly about how mutations in RB are usually associated with a poor clinical prognosis in patients with osteosarcoma. In a mouse model, we have a mouse model where we only knock out p53 in bone tissue, you see that they all get osteosarcoma and they have a survival of over 100 weeks. But if we go and mutate RB in addition to p53, now you get this much more aggressive disease. Now you start presenting metastasis, and the overall survival gets halved by the presence of that mutation. Now, this tumors also, so then the mouse model, just like we see in the human, they also have high levels of expression of UHRF1. And so we just decided to ask the question, well, if UHRF1 is important in tumor development in osteosarcoma, well, what happens if we remove it out of the equation? And so we genetically knocked out UHRF1. You can see now here in red is depicted the high expression. So these are tumors from the mouse, and the red dots are representing the high levels of UHRF1 RNA present in these tumors. And when you knock out, you see that now that the tumors don't have high level of expression that look like a wild type femur. And now this is a very busy slide. My apologies for this, but so you can see here again, this is the black line that represented the tumors with just P53 mutations. You have again the red line representing those that had RB mutations that basically survive about half. Now we remove UHRF1, so we knock out UHRF1 from the tumors from the very beginning. Now you see that the blue line basically overlaps with the black. And so the beginning, they start behaving just the same as the P53 alone. So all that increased malignancy that was brought upon by the loss of RB1 is lost. Now, at the end, there's definitely things that RB is doing that are not neutrophil dependent. At the end, you do see its separation of this line, but we see a significant increase in survival of the mice when we target UHRF1. And this is one of the first indications that maybe UHRF1, it is critical to maintain tumor formation, to get the tumors to form more quickly. But as I mentioned before, and we all know, clinical outcomes in osteosarcoma are really associated with metastasis. And so what really here caught our interest and our attention was in these mouse models that lost UHRF1. Quite intriguingly, we saw a significant decrease on the percentage of mice that formed metastasis. So the P53RB mouse that we're using here as a reference, 52% of them get distant metastasis, predominantly to the lung, but also to the liver. We remove UHRF1, only roughly 23% of the mice developed metastasis. But most importantly, those that did present with metastasis, with those metastases, at least to the lung, had significantly reduced number of mets at the same time, even though that these mice are living for much longer. So they have longer time to just grow those mets and generate more mets. Actually, what we see is that they have fewer mets and they have fewer number of metastatic nodules into the lung, which if you think about the extrapolating this out of the clinic, this is far more manageable than having nearly 4 times as many mets to the lungs. And so what we see is, and when we look at metastasis-free survival in patient samples, that we saw that, when we look back at the clinical data, we see that this is exactly true also in humans, where high levels of UHRF1 result in a lower probability of survival with metastatic-free disease. So having low levels of UHRF1 definitely is better than none. And so I'm going to spare you a lot of the in vitro analysis that we've done using human samples to show you basically, this is really takes home the message of what we're thinking we can do with UHRF1. And so we try to mimic what would happen in the clinic. So right now, we don't have any molecules to target UHRF1. And so what we've done is we have genetically modified the cells to then respond to something that we do have, which is doxycycline. So we do use this inducible knockout systems. And so we've taken human osteosarcoma cell lines, introduced we use CRISPR knockouts for our protein of interest, UHRF1, in an inducible system. So we inject cells into a mouse model, directly into the femur of the mice. And in this case, it was the flank. And this one, basically one side would get a control. So basically it would not alter UHRF1. And the other one has the ability of decrease the levels of UHRF1. And we let these tumors grow. And so once the tumors have already settled and they're visible, then we introduce to the mouse doxycycline, and that doxycycline will now trigger the knockout of UHRF1 just on one side of the mouse and not on the other. And then we track them for 2 weeks and see what happens. And what happens is this: if you leave UHRF1 alone, and so you have high levels of UHRF1, the tumors grow a lot quicker. And if you start looking also, and I'll highlight this later, but we'll talk about it, you can see that they're basically a lot redder in color. So they're highly vascularized. Whereas if the side that could knock out UHRF1 or reduce the level of the UHRF1s, you can see here, we don't completely eradicate UHRF1, but significantly decrease the levels of UHRF1. In the D-cell knockout side, you see smaller tumors and paler in color. We look at the statistics here. Overall, basically, it significantly decreases the ability of the tumors to continue to grow beyond the initial size at which we started drugging. So instead of doxycycline, instead of the genetic manipulations, this sort of indicates that if we did have molecules that could target UHRF1, then therapeutically we could result in a significant decrease in tumor growth. But most importantly, when we repeat this experiment and using an off-the-topic xenograft model, basically now we grow those osteosarcomas directly into the femur of the mice. And the beauty of this mouse model is that now we can actually we can study metastatic disease, just spontaneous mets to the lungs. What we see is that we have a significant number of mice that do not develop mets, and that those that do develop mets then now have fewer metastatic nodules. They're basically this highly dense purple-colored bits there. And here's the statistical representation where you see significant reduced number of lung metastatic nodules when you knock out UHRF1. And in terms of survival, we significantly increase survival of the mice. Now, this is completely different than we would normally see in the endpoint. So when it comes with survival here, in patients you usually will do either amputation or limb salvaging surgery. We don't do that sort of things in the mice. We basically call it an endpoint in terms of survival when the tumor has grown beyond 10% of the body weight. So basically this ethical use of animals. But you can see that basically there's a significant increase in doubling in survival while we're halving the presence of mets and even the ability of the cells or the tumors to metastasize to begin with. And I had also point out that if you look at either this flank tumors or even the orthotopic, so basically the tumors that came and were injected directly into the femur, you may see this discoloration and this indication that there's less vascularization when you are targeting UHRF1. And so we did some staining to look at vasculature. And this is quite important because vasculature to the tumor, one, it provides nutrients and allows them to grow. And also this becomes the channels for the tumors to metastasize. So less vasculature will make the tumor less likely to be able to break away and disseminate to distant organs. And you can see here, there's a lot less brown stainings that are representative of blood vessels or in the knockout compared to the controls. And basically we see that there's a significant decrease in the number of vessels in the tumors without UHRF1 than the ones that have high levels of UHRF1. While the formation, vasculature formation, in terms of the size of the vessel is not affected. This is quite important because we want to spare the rest of the body from being able to develop normal vasculature. And we can do this also in vitro. I can see here that normally lung fibroblasts are really good at inducing blood vessels in culture setting. You can see this sprouting of the cells to form blood vessels in a dish. And also sarcoma cells normally have also, not as great as lung fibroblasts, but they have this ability of guiding endothelial cells to form blood vessels. But if we knock out UHRF1 from the osteosarcoma cells, then their ability to form blood vessels is significantly decreased. And you can see in all the osteosarcoma, human osteosarcoma cell lines models, if we knock out UHRF1, so basically we remove UHRF1 from the equation, they're far less capable of bringing in blood vessels to them to generate nutrients. And so, how exactly is UHRF1 mediating this? And this is, try to then understand the pathways. We do this a little bit, one, because we're interested in the biology behind that, but perhaps for you to understand the importance of this is also, right here, right now, we have this interest in trying to drive molecules to target UHRF1, But if we understand the pathway and what else is happening for UHRF1's role, we can also identify potential other therapeutic targets that may result in the same outcome. So in this case, in terms of UHRF1's ability to induce angiogenesis, what identified is that if we look at the changes in gene expression that happen when we remove UHRF1 from osteosarcomas, you see that cell migration is one of the cluster of genes that we identify, and that brings a lot of different genes that are being altered. And one of them is the SEMA3E gene that we were able to confirm that in the absence of UHRF1, SEMA3E protein levels go up. So having high levels of SEMA3E is important. But this doesn't really tell us how it's directly being regulated by UHRF1 or how UHRF1 is resulting in dampening the levels of SEMA3E in order to aid tumorigenesis. And this is where just reading a lot of literature comes in handy. And we were quite lucky because basically the year that we were looking at this was the year that, so basically last year, there was this paper published that showed that UHRF1 interacting with this other molecule, PP2A, was able to block this AMPK. So there's a kinase that's normally regulating metabolism, but it was also regulating things epigenetically. So basically the expression of other genes through epigenetic regulation and ultimately also cell proliferation. And so this basically linked UHRF1 with AMPK, and then looking at AMPK-regulated genes through that epigenetic control, we identified that SEMA3E, the SEMA4 3E, is one of those targets. And so ultimately, we were able to validate that when we target UHRF1, that increase in SEMA3E then results, when we target UHRF1, the low levels of UHRF1 then increase phosphorylation or activation of the AMPK, and that will then result in increased levels of SEMA3E. And we can see here that if we get rid of SEMA3E, then the spreadability of the cells, of those sarcoma cells to induce sprouting is restored, which is a bad thing. So we want to make sure we preserve SEMA3E levels. So a very long story short, UHRF1 is capable, so high levels of UHRF1, which is not good for osteosarcoma, will normally be blocking the activation of AMPK, and then that will result to lower levels of SEMA3E. So might be ways of, I think those will be an induction of SEMA3E that we want here, and this blocks angiogenesis, which is a good thing. I didn't talk about this today because I talked about this other side of things during the factor meeting a few years ago, but this was published last year, so if you're interested, all the data is there. But we have also identified that UHRF1 through the expression of exosomes, and we try to understand exactly how this is part of what we're doing now. It induces or secretes plasminogen-activated urokinase, or UPA for short. And UPA can also induce migration and invasion in order to aid to metastasis. Now, one of the advantages of UPA and that could be exploited clinically is that we have tested amiloride, and amiloride uses an antidiuretic or as a diuretic, so it's commonly used in the clinic. We have using those molecules, those inhibitors of UPA significantly affect the metastatic potential of osteosarcoma cells. And there's other drugs that could be used that have not used in the clinic, but at least there are inhibitors already developed that could block this, either the production of the exosomes or the incorporation of the exosomes or the signaling through exosomes in the tumors that could ultimately also result in a decrease migration and invasion that can have effects in decreasing metastasis in UHRF1, and as well as showing that UHRF1 is affecting proliferation not only of the primary tumor, but what we see is that when we target UHRF1, also the long mets continue to divide a lot slower. And so we are convinced that UHRF1, assuming that there are not systemic toxicities associated with targeting UHRF1, we think that UHRF1 is a very attractive new therapeutic target for the treatment of UHRF1. So if we still have time, I could just basically tell you about where are we going now, basically.
Oh, can I definitely have time for that. But can I just ask you a question on that last slide? So if you're able to let's say use the amiloride on that kind of right hand side, that does that affect what happens on the left hand side with the AMPK and the SEMA3E?
No, it wouldn't. Yeah, so you would only, so this is what within UHRF1, if at all possible, if targeting UHRF1 results to not be toxic for other tissue developments, because this is the other thing that we have to keep in mind with adolescents and young adults and infants when we think about this in the setting of retinoblastoma, is that you're still developing and you still, your normal, we want to make sure that the quality of life is not affected by altering tissue development elsewhere. It will seem like it shouldn't be too toxic, but until we have the molecules and to actually do these experiments, we cannot really tell. But yeah, so I think that the best target for therapeutic intervention will be UHRF1, because then you affect proliferation, you affect angiogenesis, you affect migration and invasion, ultimately resulting in decreased tumor growth and decreased tumor metastasis. Now, if we cannot get away with that, then targeting any of these things could, make the tumor less fit to metastasize.
Thank you.
So what are we doing in terms of trying to target UHRF1 directly? And I showed you briefly before this picture that basically is a schematic of the UHRF1 protein. So I mentioned there is an E3 ubiquitin, a ring-fingered ubiquitin ligase. It also has this SRA domain that is involved in DNA recognition and to mediate DNA or maintenance of DNA methylation. It's got a PhD, TTD, UBL. So this got 5 different domains through which it can do different functions interacting with different proteins. So it's quite complex. And so our thoughts of how to minimize potential toxicities is if we can really pinpoint as to which of these domains is driving this pro-tumorigenic roles, then maybe we can target just a specific function and spare the rest, so that the rest of the normal functions of UHRF1 can still occur. And so to do this, we first have started trying to identify which are the critical domains by mutating them. And the beauty of UHRF1, or not the beauty of UHRF1, but in terms of its overexpression, if we overexpress it in these normal cells that then give rise to bone tumor, they already start showing this pretumorigenic effects. So you don't really have to study this in the concept of cancer to start seeing its effect, even though we have also found similar effects in osteosarcoma. If you overexpress UHRF1, you see this increase in migration that we saw in osteosarcoma cells. And now we mutate each one of these domains. We have already identified that it's a UVL, so this proteasomal pathway involved domain that will no longer be able to increase metastasis. And so that's quite interesting. And then there's TTD domain. So basically we could spare this E3 ubiquitin ligase and everything else that is technically involved in maintenance of DNA methylation, and then just focus on targeting these 2 domains here in hopes that then that would result in altered metastatic potential. I think that's sort of where we're going into for targeted therapeutics. Then we try to, so this part of here, and it has particular complexity because it's blocking interactions with other proteins. And as scientists, our ability to block protein-protein interactions has not been too great. we're really good at inhibiting activities, which would have been this ring domain. But there have been there's advances on how to potentially very specifically alter things that are happening at the surface of proteins. I'm not going to get too much because this is a very complex event for myself. This is where collaborating with other individuals comes super handy. So I have collaborated here at UCI in the under LoopTech. And the LoopTech lab does aptamer evolution, which basically is antibodies made out of nucleic acids. So you can use either single-strand RNA or DNA, and this is just basically random sequences that then will fold. There's no real way of predicting how they're going to fold, but then they can interact with proteins. And so what we've been doing is just basically starting with a screening of trillions of aptamers, and then started doing the selection of which ones are binding specifically to each one of those domains of UHRF1. And we've finally identified some that are key hits for each one of those domains. And so since we now know that that UBL and TTD domains may be the most important ones, we'll start validation on that area. But either those aptamers in the future, when we advance our ability to get these things into the cell, they could serve as potential therapeutics, or we can tap them with fluorophores and try to use them to screen for molecules to do competition experiments. To make basically the development of aptamers as tools could streamline and simplify your ability to screen for molecules that would target UHRF1 in the way that we want. And there's, I mentioned that the RB pathway, so we've seen here in terms of the benefits of targeting UHRF1, we're independent of whether or not you have mutations in RB. There are other ways that tumors can inactivate the RB pathway. And one of those ways is through amplification of cyclin-dependent kinase 4 and 6. And for CDK4, there are inhibitors that are already in clinic, palbociclib, are quite used for many different types of cancer. And there's actually a clinical trial that has been opened for advanced sarcomas, including osteosarcoma. And we were kind of curious to determine whether palbociclib, in the context of CDK4 amplified osteosarcomas, whether it will target UHRF1 and will result in this therapeutic benefit. And what we've seen is that, palbociclib within 24 hours is very good at decreasing the levels of UHRF1, and specifically acting through preserving this repressed state so that it blocks the production of UHRF1. And then what we see here is that when you treat with palbociclib, then you sort of see a significant decrease in cell migration. But if we use the knockouts, so basically the osteosarcoma cell lines that do not have UHRF1, then palbociclib does nothing, which means that the reduction of migration that is activated by, or the blocking of migration by palbociclib is UHRF1 dependent. And now this was done in the SJSA1 cell line, which has CDK4 amplification. We repeated this in all of the other lines, and this would also, so palbociclib would only work in osteosarcomas that have amplification of CDK4. So my prediction is that probably we will see some interesting outcomes for those clinical trials that are currently in phase 2, incorporating possible palbociclib to these patients. So that is definitely a therapeutic opportunity to decrease UHRF1 and improve outcomes in patients that is available right now. And then the other approach that we'll be using, and this may move faster through the pipeline, is taking advantage of the endogenous degradation systems that exist in the cell. I've been talking about how RB and RB pathway and CDK4, those are all involved. So this pathway here, it's involved in the production of UHRF1. But within the cell, there's also pathways that are involved in decreasing the levels of protein, right? So there's the making, and then there's also the breaking. And when it comes to breaking down UHRF1, the breakdown of UHRF1 happens through a process that's called ubiquitination. And so ubiquitination of UHRF1 will then target this protein for degradation through the proteasome. And this will normally be activated in the cell, but the cell also has the ability to block the degradation of proteins. And the blocker of the degradation for UHRF1 is a protein called USP7. A deubiquitinase. Basically, USP7 will remove the ubiquitin tags and will prevent UHRF1 to be degraded. But there's already inhibitors that have been developed to block USP7, not in the context of osteosarcoma, but they've been developed in the context of tumors that frequently have inactivation of p53 through routes that do not involve mutating the protein. And so this has been developed as a mechanism of maintaining p53 levels high in cancers in order to trigger them for cell death. But there have been some evidence showing that maybe they would also work in p53 null tumors, like in osteosarcoma. And so we wanted to see whether blocking USP7 will then result in blocking in allowing UHRF1 to get degraded through the proteasome. And so I don't have a lot of experiments done in osteosarcoma to show you, but, just kind of like basically expand a little bit the horizons, that there are things that we can test in other cancers and then repurpose them and bring them back into osteosarcoma. And so we've done the experiment of testing whether when we knock out USP7, and you can see now the loss of, so this is in triple negative breast cancer. The clinical presentation of triple negative breast cancer is quite similar to osteosarcoma. You have loss of p53, you have frequent mutations of RB1, and they have high levels of UHRF1. So this is why we use this tumors. And there's a lot more patients with triple negative breast cancer. So we can develop things and drive them to the clinic, get them approved if they work, and then repurpose them, bring them back into being used for osteosarcoma patients. So you see high levels of UHRF1, you see the presence of USP7, and we knock out USP7. Now you see the decrease of UHRF1. And now if we look at mouse models of the disease, using this USP7 knockouts, you see that now these tumors barely even grow compared to the controls. And most importantly, triple negative breast cancer is also super metastatic. You see in these mouse models, you see liver, lymph node, brain, and lung metastasis. And when we target USP7, you don't see distant mets. You only see a very low percentage of regional mets into the lymph nodes. So basically, similar to what we have seen with UHRF1, targeting USP7 significantly reduces metastatic potential. So this is using genetic inhibition of USP7. And so then we have started now, and this is fresh out of the oven. We got these results earlier this week. Tuesday was when my grad student gave me this. But we've done a short preclinical trial using the inhibitor of USP7 that's called FT671. Very small numbers, very strong drug. So we just wanted to see whether this will work or not. And you can see now, basically, we inject the tumors, allow the tumors to form. Once the tumors have reached a certain size, then we start drugging either with the vehicle as placebo control or with the drug. And now you can see that by the end of the 21 days that we tested this, there's a significant decrease in the ability of the cells to grow the tumors. And there's no toxicities associated. As you can see by the change in the body weight. We've also done blood counts. And so there's no toxicity. So this becomes something quite interesting to indirectly target UHRF1 through inhibiting the deubiquitinase that prevents UHRF1 degradation. And so with that, basically, I just want to emphasize this is work that is not necessarily done by myself, but by my team of fantastic individuals. This last freshman of the development work was done by my grad student, Ashley Kim, but also everyone has contributed in some way, directly and indirectly, to the work. But was most of the work in osteosarcoma was mainly driven by my grad student, a former grad student here, Stephanie Wu, who's now a postdoc at St. Jude. Now with that, I'm happy to answer any questions that you may have.
Thanks so much, Dr. Benavente. I think we do have a few questions from the audience that Vicky and Walker were going to take.
Yeah, I have one question to start. Is there a drug in development to directly target the UHRF1?
So to our knowledge, not yet. It is. We need to really understand. So I know that there have been different groups that identify in colon cancer that the SRA domain would be the critical one to be going after. They might be there's nothing that has been published. They might be in the process of developing inhibitors for UHRF1 into particularly that domain. But yeah, as of now, there's nothing that it's in development that is public knowledge.
Thank you. The next question, I'm hoping I pronounced this correctly. Do you think there may be a role for maintenance palbociclib to decrease the risk of relapse?
So can you repeat the question? Is it?
I can say
The maintenance of what?
the palbociclib.
Yeah.
Yeah, I'm sorry. Yeah, so do you think there may be a role for it in maintenance to decrease the risk of relapse?
I think that so for the CDK4 amplified tumors, I think that it might be of interest to look into palbociclib. If you don't have CDK4 amplification, then the mechanism that is resulting in high levels of UHRF1 might be different, and therefore you may not necessarily affect. But it might be. So in any osteosarcoma where there is either amplification of CDK4 or high or are not RB1 mutant, but have high levels of UHRF1, I think that palbociclib is a very attractive option that we have available at this moment.
Oh, right. And have VEGF inhibitors been explored in osteosarcoma? Someone else asked.
That's a good question. I do not know. Yeah, so for those that don't know, so VEGF, it's basically a vascular epithelial growth factor. So it aids in the growth of blood vessels that will be feeding into osteosarcoma. I have not seen that as a therapeutic application.
Dr. Benavente, I had a couple questions about that last slide you were showing us about potentially blocking, inhibiting USP7, kind of blocking the degradation of UHRF1.
Mm-hmm.
Yeah. And so is that, I mean, I know ideally you would just inhibit UHRF1, but I'm wondering if you can possibly do a head-to-head comparison. I'm just curious how blocking USP7, how that compares to blocking.
Well, so yeah, that's a great question. We haven't done it. We are in the process of doing this in osteosarcoma. We have done it in the triple net breast cancer background. And much to our surprise, USP7. So when we do the experiments in vitro, UHRF1 is a far superior target than USP7. When we move into the mouse, which is, I think it's a lot more clinically relevant, USP7 is amazing. So there might be definitely things that USP7 is doing, controlling beyond UHRF1 that are quite beneficial for the tumor. And so targeting USP7, it's better. And so I show you that tumor growth with the knockout that basically is flatlined. The tumors barely even grew. When we knock out UHRF1, they do grow a lot. I mean, they grow significantly more than the USP7. They don't grow as much as the vector control. And yeah, so USP target and USP7 was a lot better at decreasing tumor growth and decreasing the rate of metastasis, which is why we're now wanting to go repeat this in osteosarcoma to determine whether this would also hold true in an osteosarcoma model, because we have done the experiments in vitro, and in vitro, it seemed like UHRF1 was better, but here in vivo, we definitely have to do the in vivo experiments, and they're in the process. So, yeah.
Is there a USP7 inhibitor in development?
There are. That's a great question. So there's a lot of very good inhibitors out there. They're highly selective. If you look at them, they're phenomenal on paper. And the big question that I have and my collaborators have is why they have not moved into the clinic. Some ideas is that they haven't really identified the best tumors to move things forward. And some of the people fear that there have been toxicities, which is why we wanted to make sure that run that 21-day trial and see what were the toxicities that we observed. We didn't see any toxicities. Now, they have been developed in the context of trying to reactivate p53. So it could be that the tumors that were chosen to first run trials, they didn't work so great, and the concentration at which it needed to be used was much higher, and that we're being able to get away with a lot less dosing, and that may be less toxic. But yeah, that's something that puzzles me as to why they have not been developed. We had not been behind the use of these drugs. We just basically are repurposing them. But yeah, I'm not hopeful.
And you mentioned some other cancers. I mean, I think it is such great to be able to study this in cancers where there's just also more patients, right? So there's, and so you had mentioned the study in breast cancer. What are other cancers where you do see this high expression of UHRF1?
UHRF1. So the other one that we have personally been looking and going after is small cell lung cancer. Yeah, so small cell lung cancer. It also has, so there the opportunity to drag things into the clinic is that survival rates are 7%. So they're very aggressive tumors. It afflicts people at a much later age. So we're talking about patients that are over 75 years old. So being able to push clinical trials, much larger population, much more tractable to try to test for toxicities and such. But we see similar. So with small cell lung cancer, where we knock out UHRF1 on the mouse models, with exactly the same genetic presentation of the osteosarcoma, yeah, we see doubling of survival. So I think we have definitely identified adult cancers that were great for testing. If we come up with inhibitors, that will be great settings to test their use in the clinic. And if they work, then bring them back to childhood cancers.
Thank you so much, Dr. Benavente. Wonderful presentation. Really appreciate having you on today. Thank you for joining us on OsteoBytes and for making it better for sarcoma patients. More information on this and all OsteoBytes can be found on YouTube, on our website at mibagents.org, and also at your favorite podcast place. And next week, instead of OsteoBytes, we're co-hosting a webinar with our friends at the Raleigh Foundation and several other pediatric cancer foundations about funding opportunities from the Department of Defense Medical Research Program for Pediatric and AYA cancers. This webinar is open to scientists and researchers, and DoD Program Manager Dr. Donna Kimbark will lead a webinar on best practices and tips for applying for the Department of Defense peer-reviewed cancer research program. Last year in 2022, $56 million in funding was awarded. 44 awards were given for 9 specific cancers. So great funding opportunity. And that webinar will take place Thursday, April 6th, so next Thursday at 12 Eastern. So a slightly different time from our normal OsteoBytes. And to register for the webinar, please email amy@rileyfoundation.org, and I've put that email in the chat. You can find our OsteoBytes lineup for the next few months on our website. And if you have any ideas for future topics you'd like to hear about, please feel free to email us at events@mibagents.org. Thanks again, Dr. Benavente, Walker, and Vicki for spending an hour with us today, and to everyone else for joining us. And we hope to see you next Thursday at the DoD webinar.